I love working with the clinicians, but yeah, almost exclusively they just say Group B strep. It's Group B strep. All right. Yeah. Again, it is Group B strep. Can't argue that point. But it's Streptococcus agalactiae. Just like strep pyogenes is the major Group A strep that causes human disease. Group B strep, Streptococcus agalactiae, is really the only Group B strep that causes any human disease. So I can appreciate oral shorthand, but again, let's be proper. All right, that would be like me anyway. Stay on point, Teresa. All right, so again, high level of colonization, especially in the vagina and in the oral cavity. Why am I really focusing on the vaginal area of women today? Because here, just like we talked about for Staphylococcus agalactiae and cystitis, here it is now that transmission from pregnant women who are colonized in the vagina when they either during pregnancy, but it most often occurs during birth. If they are colonized, that baby is delivered vaginally, that baby gets infected. Mom doesn't have a problem. Maybe she develops a UTI, urinary tract infection. But typically, mom doesn't have a problem. This is all about the baby, all right? Because if that baby is infected during delivery, so this is an early onset disease. So if the baby presents with the symptoms within 7 days, you know the infection occurred during delivery, all right? We've got meningitis, we've got pneumonia, we've got some really serious infections with high rates of mortality, okay? Late onset disease, so if symptoms occur after 7 days, that's going to be the baby's infected by everybody coming in and loving on that brand new baby, right? Because everybody, or not everybody, but about half of us are carriers in the oropharynx, as well as the genitourinary. I don't know about men, but at least in the vagina of women. But everybody is, or a lot of people are colonized in the oropharynx. Now you cough, you sneeze, you pick up the baby becomes infected. Okay. Still have the same disease processes that can occur, especially with the meningitis, not as much pneumonia for whatever reason, with very high levels of fatality if this can happen. All right, how do we prevent this? Yeah, it's just getting, for the late onset, it's just making everybody, practice some really good hygiene when they're coming in to meet the brand new baby. For early onset disease, now if the women have good prenatal care, everyone is screened around 35 weeks of gestation, and if they are positive for strep agalactiae, you get an antibiotic treatment. It's really effective. We don't have any drug resistance yet, knock on wood. So you just treat mom if she's positive, and then baby is fine. Really simple, right? Yep, that's all there is to it. Virulence factors, colonization, is a recurring theme. Attachment, yep, different attachment factors, you don't need to know them, and that antiphagocytic capsule. Big thing about strep agalactiae, vaginal colonization, infecting the neonate, and screening mom at that 35 weeks. All right, here is your biochemical signature. Yeah, I'll go ahead and give you this one. I'm not going to ask you this on this exam. Do you need to know it? Eventually, yeah, if you depending on what you end up doing. But I am going to take just a second because we haven't talked about CAMP positive. And so what I mean by CAMP positive, so this is a really neat effect. So you draw this line down the middle, that is Staph aureus. Remember, it's beta hemolytic. Now here going at a perpendicular angle, you now streak with whatever bacterium that you are testing, okay? See this little arrowhead? That is a synergistic effect between the hemolysin secreted by Staph aureus and hemolysin secreted, in this case, by strep agalactiae. And where there's that synergy, you see enhanced hemolysis. And as they're meeting and getting to the right concentration, it forms that arrowhead. All right. Here, no, it's not shown on that plate. Sorry. If you look up here to the cartoon, so here's where you would have another bacterium that doesn't have CAMP activity, and you streak it, and it will grow, but there's not that arrowhead of enhanced lysis. All right, so whenever I talk about it being CAMP positive, maybe remember this arrow, and I'll maybe show you the image. All right, last group of strep. So this is a group. The Streptococcus viridans, you might see it referred to in literature that way. Viridans is not the species name, okay? It is viridans, Latin, viridans, birdie, green color. These are all alpha hemolytic, or most of them are alpha hemolytic, right? Some are variable, so there's a little bit of alpha, a little bit of gamma, depending on different conditions. But that's the basis of the name, okay? So lots of different ones, but it's that alpha hemolysis because it's turning that blood agar green. So it's verdant in color, verdant like the grass in the spring, not right now, all right? Lots of these are colonizing the GI tract, especially the oropharynx. A lot of these are oral bacteria that are causing dental caries, dental cavities, causing gingivitis, causing oral diseases. One of them, and so this one is now some of the textbooks, some of the literatures are pulling Streptococcus galolyticus out of the viridans group because really all of these other ones are in the oropharynx. Streptococcus galolyticus is in the GI tract, so it's in the colon. All right, really interesting. We'll get to that in just a minute, where it causes a lot of disease. All right. So shorthand, lots of times you'll see it. The case description, the answer: Streptococcus viridans. Oh, sorry, that's soft enough that hopefully won't be picked up. But start remembering the names. So some of the most common ones: Streptococcus mutans, immitis, mitis. Because now on all sorts of different exams, the level of precision, they're not just saying it's a Streptococcus viridans, but they're actually expecting that you will know the species name, okay? All right, so if we're thinking about the oral strep, so yes, all of those oral diseases and like the gingivitis, like causing, this is, because these form biofilms, so now you've got the cavities that can occur. But now, if you've got some periodontal disease, those bacteria in your oropharynx can get into your bloodstream. Okay, they're in your bloodstream, they can go anywhere in the body. Not as common as you see with Staph aureus or Staph epi, but a very common cause of endocarditis. So remember, bacteria growing on the heart are these Streptococcus viridans because they get into the bloodstream, now they set down on the heart, cause that vegetation. All right, but again, that's going to be part of the patient description in that case would be someone who has bad oral hygiene. They have some dental disease or periodontal disease. All right, so that's the risk factor for developing streptococcal endocarditis. Strep galolyticus, as I said, is part of the normal flora, or at least a commensal in the GI tract. But it's also now a good way that some people are diagnosed with colon cancer, because if there's a perforation in the GI tract from that cancer, from that procedure, now strep galolyticus can get into the bloodstream, can cause endocarditis, can be found in the blood. So if you go in and you have a blood culture done and the lab finds strep galolyticus, you're going to be screened for colon cancer, because that colon cancer, still, you don't know that you've got it yet, but it has perforated the bowel, and that allows the strep galolyticus to get into the blood. So just kind of a really neat story. I would not test you on that because it's not esoteric, but because it's real. That's the way a significant number of people can have been diagnosed, but it's just kind of really neat. All right, virulence factors, again, polysaccharide capsule, and being colonized with those, and then for strep galolyticus, the adhesion molecules that will hold them there into the GI tract so that they don't get secreted with the fecal matter. Diagnosis, it really all depends. All of these are associated with causing disease when there are underlying medical conditions that now allow them to get out of the oropharynx or the GI tract. So that's going to be really specific. All right, last coccus real quick. So the enterococcus. So they used to be classified as streptococci because they're gram-positive coccus. But that once we had molecular biology, we realized that there's really enough difference that a new genus was created, and it is the enterococci. Really, only the vast majority of disease are caused by two species, Enterococcus faecalis and Enterococcus faecium. Kind of picked up on something there in the names, fecal, kind of. So here, colonizing the GI tract, highly associated with not diarrheal disease, not GI disease, but now you've got transmission from the colon, from the GI tract to other parts of the body, most commonly to the urinary tract, and so highly associated with urinary tract infections. Okay, virulence factors, they're great at forming biofilm, so that helps them stick. Number one, to the GI tract. Number two, now to the urinary tract or to that urinary catheter, because I'll skip down and come back to these next 2 points. Because a major risk factor for having clinical disease from these is frequent hospitalization, staying in a long-term care facility, because that and having that indwelling urinary catheter, because now you can get colonized. Now, if you're not already colonized, or you can have transmission, but then you've got that urinary catheter where the bacteria can grow, and now you've got an inoculation. Or if there's not good care, changing that urinary catheter in the right way, frequently enough, now, because it's all kind of right there, same geography, you've got contamination from the rectum into the urinary tract, and now you've got that urinary tract infection. All right. Major virulence factor is that encoded in the bacterial genome, you have resistance genes for both penicillin and for nearly all of the cephalosporins. So a natural resistance. But we also now have acquired resistance, and we'll get to that in a couple slides. All right. So this is a major problem in treating these bacteria because of the high levels of resistance. All right. So major diseases, as I said, you're going to have the urinary tract infections. Overwhelmingly, that is the primary disease associated with enterococcus. Can be some other things, especially bacteremia, but we're really going to focus on the UTIs, okay? And again, thinking because these bacteria have that natural resistance, another risk factor for now developing clinically apparent disease, healthcare associated as well, is someone who's been on antibiotic therapy, because the antibiotics are going to kill everything else, but not kill the enterococcus, and so now you've got that easier transmission. Okay. Diagnosis, forget that for right now. Too many other things to ask about. I could ask 50 questions and not just the 10 for this lecture, but there it is. These two lectures together. But the important thing about once you diagnose that is that you must do antibiotic sensitivity testing. We already know that there's penicillin and cephalosporin resistance. The other thing, this is a major challenge, is that there is resistance to vancomycin. If you don't know this, not testable, but just because vancomycin is going to come up again and again, vancomycin is it's not quite a last ditch. It used to be our last ditch, but when there's resistance to a lot of other things, you use vancomycin. Why do we not just use that first line? Because of the side effects. So it's kind of not quite a drug of last resort, but it's almost last resort. Well, now you've got high levels of vancomycin resistance. So high that for as long as I've been studying microbiology, which has been a decade or so, we've got what we call VRE, vancomycin resistant enterococci. That's how prominent the level of vanc resistance is in enterococcus. Right? So it can be really difficult to treat. Okay, ready to start talk quickly about the bacilli. First one up, Corynebacterium diphtheria. So the last, I think we've got 4 that we're going to talk about in 8 minutes. We've got they're all bacilli. So this is a gram-positive bacilli, Bacillus arad. But what is really two unusual things about the morphology. One, see kind of this spotted, beaded appearance. I'll skip down to the bullet 2. Those are the metachromatic granules. So if you would look under a microscope to be able to see those and really focus up and down, they're multicolored and it kind of sparkles a little bit. All right? Those are specific proteins that are made in the bacterium. This is not a result of staining like we talked about with E. coli growing it on E. and B. This is something that the bacteria does on its own. All right? The other factor is that some of the bacteria have this club shape on the end. This is not a spore. Okay? This is just the morphology. The bacterium naturally grow that kind of drumstick club-shaped morphology, again, on their own for whatever reason. Don't know. Okay, if we think of transmission here, it's going to be those respiratory droplets because the site of infection is here in the oropharynx. Diphtheria is the classic presentation. Patient's going to come in with a really sore throat, and you look, and there's going to be enlarged tonsils with a grayish-white membrane that covers them. You try and remove that membrane when you're doing your throat swab, and if you move it, it tears and it bleeds. It's firmly attached. Okay, that is the classic presentation. There's a couple other things we'll see on the slides, but that is your classic hallmark signs of diphtheria. All right. Important thing: DTP, D in your DTaP vaccine is diphtheria. So we can prevent this with vaccination. So now when you see those patients, typically they're patients who either don't believe in vaccine or their parents don't believe in vaccine. They have not been vaccinated for whatever reason. Immigration, vaccine hesitancy, those are the kind of the populations that we're thinking of. Okay. Identification, you're going to look for that morphology, and then if you plate them on this specific agar, you see those black colonies develop. Okay. That diphtheria, the D is diphtheria toxin. That's the major virulence factor. So the toxin is one of our classic AB toxins. All right. It is mechanism of action here. Remember the AB. A is the adhesion, is the activity domain. B is your binding domain. So you have these two domains. B binds to the receptor, triggers internalization. A separates and then goes and does whatever A is supposed to do. In diphtheria toxin, that A subunit binds to the elongation factor. So think about you go back to your genetics course with Dr. Verghese. Think about translation, where the elongation factor helps in your translation process. Now, if that diphtheria toxin binds to it, EF2 can't do its job. It's kind of it's just steric hindrance. It can't process through the translation molecule or the translation process, and so now protein synthesis stops. Protein synthesis stops, the cell dies. There's your toxic activity. All right. I'll go ahead and give you this one. I'm giving you lots of clues today. All right. So yes, when we start talking about all the gram negatives next week, you're going to have a lot of AB toxins that you will need to know the mechanism of actions. Don't need, yes, eventually someday maybe you'll need to know this one. Don't worry about it for Monday. All right. But this is just an example of what we're going to talk about a lot in the very, very near future. Okay. Here, major disease, as I said, is diphtheria. So here is that grayish membrane that is covering the tonsils. And see how red and inflamed the oropharynx and the tonsils are there. Okay. You also now have this bull neck. So see the inflammation that's occurring. This is not a swollen cervical, an enlarged cervical lymph node. This is not lymphadenopathy. This is now the toxin that is causing inflammation in the tissue, not in the lymph node. But that is, again, a very prominent hallmark sign of diphtheria, that bull neck. Think about the bull neck. Okay? You can have some other diseases as the toxin, so major cause myocarditis. So the myocardium, the sac covering the heart, itis, inflammation. The toxin can now disseminate, get into the bloodstream, disseminate to the heart and cause inflammation there. And then you've got some other diseases that we won't focus on because of time. Just focus on the classic presentation there of fairness. And yes, prevention with that DTaP vaccine, where the only part of diphtheria in there is that diphtheria toxin, but it's made into that toxoid. All right, Listeria monocytogenes. Okay, gram-positive bacillus. This is the one I referred to at the beginning with the question of the first hour with the question about growing it cold, and everybody knows about Listeria now, right? Boar's Head had the Listeria. Boar's Head, that's all deli meats and cheeses. That's kept cold. Why was that a concern? Because Listeria monocytogenes grows really well at 4 degrees and grows really well in the presence of salt. Yeah, think about all that great lunch meat and cheeses. Yeah, kind of salty. Yeah, you can talk to Dr. Wilson about whether you should eat it or not for a different reason, but that's why the bacterium can grow, why it was that source of the outbreak. And it's not just deli meats and cheeses. It is many other products. All right, so it grows really well at 4 degrees and high salt. Okay? It's in a lot of different places, so that's how it gets into the meats, how it gets into the cheeses, how it gets onto those lettuces, onto those alfalfa sprouts. Major outbreak about 10 years ago was alfalfa sprouts, because all the vegetarians were like, I'm not eating meat, so I'm not getting E. coli, I'm not getting Listeria, because I'm not doing dairy. Well, where does all the fecal matter from those animals go? And now it gets onto all of that organic produce, and it can grow because you're using organic processes and not all the chemical fertilizers or insecticides, and there's the transmission there. So yeah, really interesting ways to think about it. Okay, so now how do you get it? That eating the wrong foods. But mom gets infected. This is why pregnant women are told not to eat the soft cheeses like Breeze, not to eat raw dairy during pregnancy, because dairy is one of the highest associations with Listeria. You're milking the cow, or the machines are milking the cow right there near the rectum area, and the milk gets contaminated. Then you make the cheeses. It's not pasteurized, so it's raw. Now mom eats that, gets infected. Mom doesn't really get sick. It's the baby that gets sick with serious disease, because this can be transferred transplacentally across the placenta, and it can happen the baby can be inoculated during birth. Okay? So really high risk, neonates, pregnant women, not necessarily because of their disease, but because of transmission, and then the elderly, and anyone with cell-mediated immune deficits. Okay? We've got that camp activity, so here's another good example. So there's the camp positive, there's the camp negative that we talked about a few minutes ago. And then you grow this, and you've got tumbling motility. So it's like doing cartwheels. And so you put the bacteria on the slide, and as long as it's not fixed, it's got a little bit of media, you'll see that it's spinning like a top. And you can see this, watch that video. Okay. So virulence factors here, this is an intracellular infection. It's getting inside the macrophages. And we've got several different proteins that mediate that you don't need to know. But now it's endocitosed. It's phagocytosed. So it's in a phagolysosome. That's a bad place to be, right? Because if that phagosome fuses with the lysosome, there's going to be killing. How does the bacteria survive? It expresses a specific protein, Listeria lysine O, that now pops a hole in the phagosome, and that allows escape. And that's how now that bacterium can set up that intracellular infection there in the cytoplasm of the macrophage and grow just fine inside that macrophage. Okay? The next really neat thing that it does, it expresses a different protein, ACT-A. ACT because it works on the actin cytoskeleton of that macrophage. And you can see that here in this immunofluorescent stain. Here in red are the bacteria, here in this cartoon, because that ACT-A protein, by design, rearranges the actin cytoskeleton and forms a rocket at the polar end of that bacillus that now propels the bacterium from this infected cell to its uninfected neighbor. I mean, it just basically turns it into a rocket that shoots it across that cell barrier. And so you can see it in the cartoon, and you can see it happening here in these infected cells. Okay? So 3 major virulence factors: intracellular infection, expression of Listeria lysine O, expression of ACT-A, but you can really think more about the formations of those rocket tails. All right. The diseases in a healthy adult. So lots of times we're asymptomatic, or it's just like, you know, I've got a little bit of indigestion. Maybe it's not so much indigestion. Maybe it's that you had that raw dairy, that brie, or raw milk. In pregnant women, so most often the pregnant mom who eats the food has the gastroenteritis, but that's about it. She doesn't develop more severe disease. What we need to think about, but what the most serious outcome from her is now if the baby gets infected and she loses the pregnancy, so that's spontaneous abortion. Okay. Who we really need to think about are people who are either older or immune suppressed because of those cell-mediated deficiencies, and the neonates. So here they don't have a good functional immune response, and so now they can't combat that infection. And now instead of having that gastroenteritis and some flu-like illness, they're going to develop meningitis. All right. Whether it is the older patient, whether it is the neonate, the meningitis is one of the most common, as well as now this granulomatous infantiseptica, which is now you've got disseminated listeria growing everywhere throughout the body, presents as abscesses, but don't think about that. But so again, if you want to think about severe disease and listeria, it's meningitis. And that's what you're seeing in all of these food outbreaks. So for most patients, it's the enteritis, but when you're talking about people dying, it's your older patients, it's people who are immunosuppressed that are dying from the meningitis. Okay. All right, so Bacillus cereus, I'll take 1 minute to point out the major things about Bacillus cereus, about the two bacillus. So for Bacillus cereus, you have two different toxins. One that is heat stable, so really strong association here is fried rice. The bacillus, the spores, get contaminated with any of the grains in the field, but especially rice. How do you make fried rice? You boil the rice, then you let it you cool it off, let it set on the back of the stove until it's cool, then you put it in your wok or whatever, and stir it real quickly, right? Everybody makes fried rice, that's how you do it. Okay, here's the key. While it's setting back there on the back of the stove cooling off, the spores, which were not killed just by boiling, now those spores germinate in that rich environment, and they start making a toxin. That toxin is heat stable. You put it in the wok, that toxin isn't going to be natured. Now you eat your fried rice, and 3 to 6 hours later, you're throwing up. Okay? That's the classic presentation. All right. The second toxin that's made is heat stable. That's what happens, strongest associations, meats, gravies. Now the bacteria, the spores survive the initial cooking, you eat the food, the spores get into your intestinal tract, the spores germinate in your intestinal tract. Here's the diarrheal form. They get into the intestinal tract, sporulate, and start making that second toxin, which is heat labeled. If it were outside the body and you heated it up, the toxin would be inactivated, but it's not. It's all in your intestinal tract. And now it's making the toxin, and now you have diarrhea. So same organism, two different toxins, two completely different presentations. With the first toxin, you're only going to be vomiting. You're not going to have diarrhea. With the second toxin, you're only going to have diarrhea. You're not going to be vomiting. All based on different toxins, different locations. One you're consuming, one the toxin, because the spore is in the rice. The other you're consuming the spore that makes the toxin in the body. Okay? That means you've got different time presentations. Presentation for the emetic, the throw-up version, 1 to 6 hours. Presentation of the diarrheal form, at least 6 to 12. It can be up to 24, 48 hours. Because the toxin, the bacteria has to germinate, has to make the toxin, and then you have to have the disease. Okay? All right. Anthrax, you've got spores that now have this boxcar morphology. Bioterrorism. If I say anthrax, what do you typically think of? Inhalation, everybody has the respiratory disease, right? Okay, that is the bioterrorism form. That can happen. That is the most severe form. Okay, I'll come back to the slide in just a second. But the most common form is actually skin disease, okay? Because this is, the bacillus is zoonotic, so the anthrax. So we're thinking about now the sheep, the cows, especially sheep, that are making the bacteria. The spores get onto the skin of that, the shepherd, the cattle herd, and now you've got these lesions. And you have the classic presentation, this big open ulcer with that necrotic lesion as it develops. Okay, it's called black eschar. Right? That is the most common form. But everybody thinks about pulmonary anthrax, which fortunately is more rare. Here you're inhaling the spores get into the lung, germinate, and produce the toxin. And I'll go back a couple slides to cover the toxin. Okay? This is inhalation anthrax, wool sorter's disease, because think about shearing sheep, you're there pulling the wool apart, and you're now releasing all the spores from the wool, and you're inhaling that. So they're still an occupational. Signs of anthrax. Classic sign is it's called a widened mediastinum. So this is a normal chest x-ray. Here's the spine, the trachea, sort of showing on top of each other. Here's the heart. Everybody kind of see that? Right. Here is your lungs on anthrax. All of this widening all the way down. This is not, oh, well, this patient just has a bigger spine, has a bigger heart. No, this is inflammation. All of those lymph nodes, especially the mediastinal lymph nodes, but you have this little chain of lymph nodes that go down the center of your respiratory tract. This is where the Bacillus anthrax goes and lives, causes massive inflammation, and that's what you see. This is all areas of inflammation there along your mediastinum, there in the center of your respiratory tract. That is pulmonary anthrax. Very often fatal. All right, so if you think about the toxin, you've got your major virulence factors, spores. This is the one bacterium that I talked about earlier where it does have a capsule that does have polysaccharide. Does not have polysaccharide. It is completely a polypeptide. This polyD-glutamic acid. That's 100% of the bacterial capsule. This is the one exception to the bacteria capsule being polysaccharide. Okay. Then the third major virulence factor is the toxin. So this is kind of a variation on the classic AB toxin. It is an AB, but there are two A subunits. The B subunit is the protective antigen. The two different A are the lethal factor and the edema factor. And they compare together. Got a better picture on the next slide. So here where you have in the purple, you have multiple subunits of the protective antigen, the PA, and it can bind either or both of the lethal factor and the and the edema factor, the LT and the ET, okay, or EF, sorry. All right, and now that is internalized here because the PA, the binding moiety, is bound to the receptor. That is internalized, and now the EF, the LF, the lethal factor, the edema factor are released and then go do different things. Okay, that is now causing massive inflammation, causing all the toxic activity. Okay. So technically, it's not an AB, but in my mind, it's easy to think about it as a variation on that theme because it's working in the same way. There's just two A subunits. All right, really quickly, and I went over, but these two are really important as far as the two bacillus organisms. All right, I won't ask for questions, bring them tomorrow, and I'll get out of the way for Dr. Wilson to come in. And you can ask her